Today, we will take you to explore a new concept that is, for more than 60 years we have been following. DNA transmits information through RNA, which ultimately determines the synthesis of proteins, but proposed a more comprehensive and dynamic theory of life information transmission. This groundbreaking research tells us that the information flow of life is far more complex than imagined. In this video, we will interpret with you the scientific evolution from to cellular dogma reshapes our understanding of genes, cells, and even the essence of life ***. Let us uncover the ultimate mystery of life information together. In 1958, Francis Crick proposed a groundbreaking concept that revolutionized our understanding of biology, the central dogma. This elegant idea posited that genetic information flows unidirectionally from DNA to RNA to protein. DNA, the molecule of heredity, served as the blueprint of life. This blueprint, encoded within the sequence of DNA's four-letter alphabet, held the instructions for building and maintaining an organism. The information within DNA is transcribed into RNA, a messenger molecule that carries the genetic code from the nucleus to the ribosomes, the protein synthesis machinery of the cell. Ribosomes translate the RNA message into proteins, the workhorses of the cell responsible for a myriad of functions. From catalyzing biochemical reactions to transporting molecules across cell membranes, proteins are the building blocks and the functional units of life. The central dogma provided a powerful framework for understanding the flow of genetic information. However, as with any scientific model, new discoveries revealed its limitations. Scientists began to uncover exceptions to the unidirectional flow of information proposed by Crick. Retroviruses, such as HIV, challenged the central dogma by demonstrating the reverse flow of genetic information. These viruses possess a unique enzyme called reverse transcriptase, which allows them to synthesize DNA from an RNA template. Another group of viruses, including the infamous SARS-CoV-2, the causative agent of the COVID-19 pandemic, challenged the DNA-centric view of the central dogma. These RNA viruses lack DNA entirely and replicate their genetic material using RNA-dependent RNA polymerases. Furthermore, the discovery of epigenetic modifications added another layer of complexity to the central dogma. Epigenetics refers to heritable changes in gene expression that occur without alterations to the underlying DNA sequence. The limitations of the central dogma, coupled with an explosion of new discoveries in molecular biology, paved the way for a new paradigm, the cellular dogma. Proposed by Stephen R. Quake in 2024, the cellular dogma recognizes the cell as a complex, interconnected network of information flow. This new dogma shifts the focus from a gene-centric view to a cell-centric perspective. It acknowledges that information flow within a cell is not limited to DNA, RNA, and proteins, but encompasses a diverse array of molecules and processes. The cellular dogma incorporates epigenetic modifications, non-coding RNAs, protein modifications, and cellular metabolites as integral players in the intricate web of cellular information. It recognizes that the cell is not merely a passive recipient of genetic instructions, but an active interpreter and responder to its environment. The cellular dogma expands the language of life beyond the traditional trio of DNA, RNA, and proteins. It recognizes that information within a cell is stored, transmitted, and interpreted through a diverse array of molecules. Non-coding RNAs, once dismissed as junk DNA, play a crucial role in regulating gene expression. These RNA molecules, including microRNAs and long non-coding RNAs, can silence or enhance the translation of specific messenger RNAs. Protein modifications such as phosphorylation, acetylation, and methylation act as molecular switches that can alter protein activity, localization, and interactions. These modifications add another layer of complexity to the proteome, the entire set of proteins within a cell. Cellular metabolites, the small molecules that are the intermediates and products of metabolism, are no longer viewed as mere bystanders in the cellular symphony. They act as signaling molecules influencing gene expression, enzyme activity, and cellular responses to environmental cues. Section 5. A multidimensional flow information dynamics in the cellular dogma. Unlike the unidirectional flow proposed by the central dogma, the cellular dogma embraces the complexity of information flow within a cell. It recognizes that information flows in multiple directions, forming intricate feedback loops and regulatory networks. Gene expression is not solely determined by the DNA sequence, but is influenced by a multitude of factors, including epigenetic modifications, non-coding RNAs, and cellular metabolites. These factors can act as switches, amplifiers, or dampeners, fine-tuning gene expression in response to internal and external cues. The cellular dogma acknowledges the dynamic interplay between different cellular compartments. The nucleus, once considered the sole repository of genetic information, is now seen as one node in a complex network of communication that includes the cytoplasm, organelles, and the extracellular environment. Section 6. From genes to cells, shifting perspectives. The transition from the central dogma to the cellular dogma represents a fundamental shift in our understanding of biology. It moves us away from a reductionist, gene-centric view towards a more holistic, cell-centric perspective. The central dogma, with its emphasis on the unidirectional flow of information from DNA to protein, inadvertently fostered a view of genes as the sole determinants of cellular behavior. The cellular dogma, in contrast, emphasizes the cell as the fundamental unit of life, capable of interpreting and responding to its environment in a dynamic and integrated manner. This shift in perspective has profound implications for how we approach biological research and medicine. It encourages us to look beyond individual genes and to consider the complex interplay of molecules and processes that govern cellular behavior. Section 7. The cellular dogma in action, understanding and treating disease. The cellular dogma provides a powerful framework for understanding the molecular basis of disease. Many diseases, including cancer, neurodegenerative disorders, and metabolic diseases, arise not from defects in single genes, but from disruptions in the intricate web of cellular information flow. Cancer, for example, is no longer viewed solely as a disease of uncontrolled cell growth, but as a breakdown in the cellular communication networks that govern cell proliferation, differentiation, and death. Mutations in genes involved in these pathways can disrupt the delicate balance of cellular signaling, leading to uncontrolled growth. The cellular dogma also sheds light on the development of drug resistance, a major challenge in treating many diseases. Cancer cells, for instance, can develop resistance to chemotherapy by altering the expression of genes involved in drug metabolism, transport, or target pathways. Section 8. The future of medicine, cellular therapies, and artificial intelligence. The cellular dogma is ushering in a new era of medicine, paving the way for innovative therapies that target the cellular information network. Cellular therapies, which involve modifying cells outside the body and then reintroducing them into the patient, hold immense promise for treating a wide range of diseases. CAR T-cell therapy, a groundbreaking immunotherapy treatment for cancer, exemplifies the power of harnessing the cellular information network. In this therapy, a patient's own T-cells, a type of immune cell, are genetically engineered to express chimeric antigen receptors, or CARs, that target specific proteins found on cancer cells. The cellular dogma is also driving the integration of artificial intelligence, or AI, into medicine. AI algorithms, with their ability to analyze vast data sets and identify complex patterns, are proving invaluable in deciphering the intricate networks of cellular information flow. Section 9. A new era of discovery, embracing the complexity of life. The cellular dogma represents a paradigm shift in biology, challenging us to embrace the complexity of life. It recognizes that the cell is not a simple machine, but a dynamic, self-organizing system with emergent properties that arise from the interactions of its many components. This new dogma is not meant to replace the central dogma, but to complement and extend it. The central dogma provided a foundational understanding of the flow of genetic information, while the cellular dogma builds upon this foundation, revealing the intricate web of interactions that govern cellular behavior. As we delve deeper into the complexities of the cellular information network, we can expect to uncover new principles of biology, leading to innovative therapies and a deeper understanding of the fundamental processes that govern life. The cellular dogma marks the beginning of a new era of discovery, one that promises to transform our understanding of life itself.